It's my pleasure to introduce today's seminar speaker, Dr. Karin Kiontke from NYU. So Karin is from Germany originally, and she was born and raised in Berlin and got her doctorate in Berlin. And what she studied for her doctorate were basically the ecology of nematodes and saguaro cactuses, actually, like these kinds of cactuses. Okay, like from
Rotting saguaro
what?
cactuses.
Oh, sorry, rotting saguaro cactuses. Okay, nematodes love rotten stuff. So, and so she came to Arizona a lot to do those studies, even though she was studying in Berlin. And then in 2001, she moved to start her postdoc at NYU in the laboratory of Dr. David Fitch. And she kind of got promoted to senior scientist in that lab recently. And she's one of the things I really admire about Karin is that, to speak like Fairfield language, she's both a Block 1 person and a Block 3 person. All right, so what she's going to talk to you about today is a really like hardcore molecular developmental biology, molecular genetics project, finding genes that are important for particular developmental events in nematodes. I'm super excited about it. But she has this like really outstanding background in evolution and ecology. She's one of the world's experts in different kinds of Caenorhabditis species, or nematode species in general, actually. And she's the belle of the ball at nematode meetings. Everyone wants to talk to her. So anyway, we're really lucky that we could convince her to come here and give a talk. So I hope you'll join me in welcoming Karin.
Thank you, Anita, for introducing me. Thanks for inviting me. What's Block 1? Am I giving a Block 1 or Block 3 talk?
For this talk?
Okay, so that's my hat for today. So I'm going to talk to you about the timing of development. So let me see whether I can do that. Oh, the first thing I have to say is that since... okay, have I turned this on? No?
There you go. You got it.
There we go. Okay. So so this is the work. The work I'm talking about here is in collaboration with a whole bunch of people. I'm going to talk about work which has been done by Antonio Herrera, who was a graduate student, is now a postdoc in the lab of my boss, David Fitch, at NYU. And then we also collaborate with these people at the University of Rochester. Edward Wong is also a graduate student, and Douglas Portman is the professor there. And Eric Schwartz has contributed to this also. And this is kind of my acknowledgement slides. I'm going to put it at the beginning because I don't want to pretend that I did this all by myself. So now let's get started. I'm going to talk about a bunch of things here. First, I will say something about the control of development. I will introduce some genes in C. elegans, which we call heterochronic, and I'll explain what that means. I will talk about the male tail of C. elegans, which is our model, which we study, and then introduce two new genes. We'll say something about what they are and how they function and why we should care about these things. So I don't know how many of you have really wondered how this can happen, that one cell is turning into a perfect multicellular animal. And if you haven't wondered about it, I suppose your mom did at one point, and maybe your dad too, because that's what happens. Imagine what happens. So there's this cell here, it's going to turn into a mouse. It will divide, but these cells also have to somehow change so that they are different from one another. They have to make organs in the right place at the right time so that when you have a fly, that the actually antennae are at the head, that the legs are on the thorax, and the copulatory organs are in the abdomen and not anywhere else. And for the positional information, the control of development in space, we know a little bit about that. We know actually quite a bit about this, and maybe you have encountered that in your class already. There are these genes, which are called Hox genes, and they determine what happens at which segment of these flies. This was discovered quite a while ago by people who got a Nobel Prize for that. They find that, for instance, this pink gene, ultrabithorax, is gonna make that segment make this third leg And it doesn't make any big wings. It makes some little halteres. Flies have only one pair of wings. If there's a mutation in ultrabithorax, this segment is going to develop like the segment before that, the second segment on the thorax, which then turns into this famous phenotype of a fly. It's the bithorax fly. It has two pairs of wings, but these are identical because it makes this one segment again. So, as I said, people got a Nobel Prize for this because it was pretty amazing that there are such genes which regulate what happens from the anterior to the posterior of this animal. This is even more relevant because it turned out that these Hox genes regulate how development happens in space, not just in flies, but also in mice and in all other animals. So that is a really general mechanism how development or the organization of an animal is controlled. But development has to be controlled only also in time, and this is where I'm coming to the subject of my talk. So another example from vertebrates, these are tadpole larvae of amphibians, and eventually they have to turn from this legless aquatic animal into, for instance, a frog. And that is known since 1912 that this is controlled by the thyroid hormone. So when thyroid hormone is present, then these tadpoles are going to start growing legs. And to note that the hind legs in frogs actually appear before the front legs. In frogs, the tail is going to go away. The cells are dying, programmed cell death. In the salamanders, the tail remains, but they have these external gills here, and those will go away during that metamorphosis. That is controlled by thyroid hormones. And hormones are also really important for controlling another really important switch or transition in all animals. Including in us, which is the juvenile to adult transition, which we call usually puberty. And that is also controlled by hormones from the hypothalamus, the pituitary gland, and from gonads. And timing is also really important. If something goes wrong in timing, then that in humans and in these vertebrates, if it happens early enough, it's going to lead to birth defects, and we actually don't even know of those defects because it's so bad. But occasionally, a change in the timing of the development can be of an advantage. For instance, in the case of this animal here, which is an axolotl, and I don't know what happened with the label of this. This is an adult of this creature, and it still has these gills. So the gills do not go away. So it has that juvenile feature as an adult, which is good because these animals can now breathe underwater. So they don't have to live on land. They can stay in the water. And such changes of the development of just one timing of development of one organ, like this here, relative of all the rest. So this is completely developed adult with gonads and everything, but it still has that juvenile feature. We call that heterochrony, and that is a word which will come up in my talk a few times. So that can be fixed in a population, as in these, and it can also lead to just mutations, which we study in animals, in our animals. So now I said that these hormones are triggering metamorphosis here and the puberty in us, but what turns the hormone production on? So what tells the hormones to come up? So the question here is there, like for the Hox gene, a dedicated molecular mechanism which is really only regulating timing? That is difficult to study in animals like this, and that's where the worm comes in. And I don't know how much I have to say about worms. Everybody seems to have seen them before. We have Anita here who says this is her favorite animal. They are really great. They are a wonderful model organism, and there is a reason that multiple people got a Nobel Prize for working in these guys. they are really small, so you can fit thousands of them into a petri dish, which is very useful. They develop fast, so this life cycle here can be completed in less than 3 days if you set the temperature to 25 degrees. Actually, they develop from this embryo to the adults through 4 larval stages, and there's a mold between each of those larval stages. And the last larva develops into an adult, which usually in C. elegans looks like this, like a female, but these are hermaphrodites. They make sperm, and they can fertilize their own eggs with the sperm. As a model organism for genetic research, this is really fantastic because you don't have to put males and females together every time. You can just let them self, as we call it. But there are also occasionally males, like 1 in every 1,000 animals is a male, which is very good because we can use those to cross different strains with each other. Another really cool thing about these worms is that they're transparent. The people who looked at them recently, right, you can see the eggs. You don't actually have to cut the worm open. You can already see the embryos through the mother, which is very cool. You can also see nuclei, you can see the gut, you can see everything. You don't have to dissect them. These animals have a fixed number of cells. So males have more cells than females. They have 1,031 cells, and females or hermaphrodites have 965, I think. Is that right?
959.
59? Okay. So I got it wrong. She's the expert, really. And another really cool thing is that these nematodes have an invariant cell lineage, which means that from the zygote to the adult, they all develop in exactly the same way. The cells divide in the same way. So people have studied this, so there are these cell lineage diagrams. It's going to be the same in every worm, which means that you can look at these divisions and see whether there are any changes in a mutant. And that has helped people to discover genes which change the timing of development. People were looking at these cells here, which are a row of 10 cells on either side of the larva when it hatches, the L1 we call those. These cells, you can see them here. They have adherence junctions around them, which you can stain, or you can stain them with a fluorescent dye, so you can see this here. Also, all of those cells divide in a particular way in each larval stage. So there's going to be one division in each larval stage. One cell is going to remain a seam cell, and the other one is going to fuse to the rest of the epidermis. Except in the second stage, when there are 2 divisions. And that's one division is actually going to make 2 seam cells, and then the rest is going to be the same again. At the last larval stage, these cells going to stop dividing, and they're going to fuse to each other. So you can see this here, there's only one long cell here. And they will also produce a particular kind of cuticle, which is only present in adults. It has these stripes on either side. I don't know, when I was younger, we had like Adidas clothes was really cool, and they had these stripes on the side, so it's kind of like that. These ali, as we call them, they are present in adults. So now there were mutants where this division was different. Like for instance, this Lin4 is a really famous gene, and I tell you later why it's famous. And here the division of the first larval stage is repeated over and over and over again. And then there's another mutant where that division of the first larva is completely skipped. And when this happens, then the ali, those stripes, appear one stage too early. So that is why it's a change in the timing, right? So you get these ali early, and we call that precocious. There's also another really famous gene, LAT7, where this last division is repeated, so that the adults actually don't have ali, but they make them later. So that is retarded. So there are a whole bunch of those mutants, and people have done a lot of work actually to figure out how these genes all work together to regulate the timing of this last thing which happens with the seam cells between the L4 and the adult stage. This is simplified here. Actually, if you would see the real thing, it would be much more complicated, but we don't have to go there. So again, at the last larval stage, these seam cells are going to change what they do. They don't divide anymore. They're going to differentiate. They exit the cell cycle, and that is, they fuse and they make these algae. And I can explain to you how these genes work together, but before I do that, I'm going to tell you why Lin4 and Let7 are so famous. Has anybody heard of microRNAs before? Yes? Okay. Lin4 and Let7 were the first microRNAs which were ever discovered. And for the people who don't remember what microRNAs are, they are these 22 nucleotide long pieces of RNA which have a regulatory function. They can bind to the 3' UTR of messenger RNA, and they will block translation. And they're actually being transcribed as something much longer. So it's a gene which has many more nucleotides than just 22, and it's cut in two places, once in the nuclear and then again in the cytoplasm to make the mature microRNA here. So when these things were discovered, people first thought, that's something strange in worms. But then later, it turned out that we have thousands of microRNAs, and worms have thousands of microRNAs, and every organism has lots and lots of these little regulatory RNAs, and that changed how we see RNAs. At first, for a long time, people were thinking that RNAs are nothing more than a messenger between the gene and a protein, right? Messenger RNA. And they were also involved in translation as tRNAs and ribosomal RNAs, but then the RNAs could have like a regulatory role, and a really important and universal regulatory role was not known. And when these microRNAs were discovered, they started the non-coding RNA revolution. It's actually called that, and so it was really, really cool that people found these two genes in worms. So let me quickly see whether I can quickly explain how these genes are working together. This is kind of complicated here. We can start from the bottom. In order for the seam cells to actually differentiate, you need a transcription factor, which is Lin29. Lin29 protein comes up at this time during the L4 stage, although the messenger RNA has been around much longer. It's earlier already there, but it's not being translated, and that is because there's Lin41, which is a protein, an RNA binding protein, which prevents translation. So now at this stage, Lin41 has to disappear, and that happens by Let7. So Let7 microRNA binds to the messenger RNA of Lin41 and prevents translation. And Let7 comes up only here, and the reason that Let7 is not on any earlier than at this stage is because there's Lin28, and that inhibits the biogenesis, the chopping of the longer RNA piece into smaller pieces so that you get the 22 nucleotide RNA. And then there's also a transcription factor here which promotes the production of Lin28, and of course Lin28 has to disappear eventually so that Let7 can come up, and that is happening partially by Lin4. So this is a progression of different levels of these proteins and RNAs, and you have to have the right level of everything for this to happen. And these genes are actually also controlling what happens during earlier stages, but we don't have to worry about this. So the reason that it is really important to think about this is also that this is not just a wormy thing. So Lin28, Let7, and Lin41 are conserved in animals, so we have them too, and they appear to regulate these developmental switches. So the juvenile to adult transition, Lin28 plays a role in puberty, the timing of puberty, but also other switches between stemness and differentiation. Like think of stem cells and differentiated cells, or in the other direction, when a differentiated cell dedifferentiates and turns into cancer. So these are important genes to understand, and this whole pathway is important to understand also for that reason. So it is useful to study this, and that's what we do. And we are looking at it in worms again, because worms are a very good model system to study this in, but instead of the hermaphrodite, we are looking at males. And this is the tail of an adult male, and it looks quite different from an hermaphrodite tail. The male needs some structures for copulation, so we find these things here, they're called rays, and there are sensory organs which the male can use to feel for the vulva and the hermaphrodite so that he can copulate with her. They have that fan, which we think works like a suction cup so that they can have a good contact to the hermaphrodite or female, and actually we don't care about any of this. In our lab, we care about the very tip of the tail, which is like a tiny little round thing here. But in larvae, it's long. And during this transition from the fourth larval stage to the adult in males, the tail changes its shape. It undergoes morphogenesis from something pointy like this to something little round like that. This tail has 4 cells, and these cells are going to retract from the cuticle. You can see this here, these are the cells, this is all the cuticle, and then they're gonna fuse together so that there's only one cell, and that one cell is gonna move further towards the head and pulling everything forward this way. And a little later, these rays are also gonna develop so that in the end you have a male tail here, and the tail tip is really small. So we were thinking in our lab, actually my boss was thinking this before I even joined the lab, that this is a nice, simple system to study morphogenesis, because all of those things have to happen in a coordinated way. He thought that maybe we're going to solve this pretty soon or quickly, and we've been working on this for, I don't know, he's been working on this for decades now, so it's not as simple as we thought. But we can study this using mutants. So, for instance, this one where this process doesn't happen properly. So you can see that the tail tip is not retracting really, and in the end, the male here has the tail tip still sticking out. And this mutant happens to be a gain-of-function mutation of this gene LIN41, which I introduced earlier. It's one of the genes in that heterochronic pathway, which is also present in humans. We have a version of it. So here, this retraction of this tail tip is happening too late. It's a retarded, excuse me, phenotype. We call this LEP. I'm sorry. A sip of water. And if we have a loss of function mutation, this retraction of the tail tip happens one stage too early, already in these L3s. And when that happens in L3s, L4s are going to have already a rounded tail tip here. And then it continues, and the adult male is going to have a way too short tail. It's like over-retracted, we call that. So apparently LIN41 is involved in the timing of this morphogenesis, and we looked whether the other genes in this pathway are also involved in this, and we found they do. LIN4 and LIN7 have retarded phenotypes. LIN14, LIN28, and LIN41 are precocious when you knock them out. I should mention that this thing here, DMD3, is a different transcription factor, which is downstream of this heterochronic pathway to make tail tip morphogenesis. Earlier, it was a gene called LIN29 for the seam cell, so there's something different downstream of LIN41, but this is regulating the timing of this process also. So this cannot be the whole story, though, because we have other mutants in the lab which are also having a retarded phenotype. So the males, when they're hatched, when they molt, they have totally unretracted tails. They're really long, but if we are looking a little later, we see that they start retracting their tail tip as adults. So that is obviously retarded because they do this as adults. They also do something really horrible for them. They will molt again. So in adults in worms, they never molt. I know that adult lobsters keep molting because they keep growing, but when worms are adult, they never molt again. These guys do this again, which is not very good for them. They're gonna die trying that. But strangely, they do not have anything wrong with their seam cells. So remember, I told you the seam cells are the structures in which all of these other genes have been first discovered, and we only found later that worm tails use them too. But these genes do not have the seam cell phenotype. So there must be something special about those genes. And it was worth looking at them. And we did this, we wanted to know where in this pathway do they act. And we did a whole bunch of genetic epistasis experiments, which I'm not going to explain here because otherwise I won't have time to tell anything else. We found using those experiments, which involve crossing different mutants and look at the phenotype, that both of those genes are upstream of Lin28. And they are both having a negative interaction with Lin28, which is already kind of obvious from the kind of phenotype. These are retarded and these are precocious mutants. So that was pretty cool. We had 2 new genes which nobody else knew about. The people working in seam cells would never find them because they don't have anything wrong with their seam cells. So what are they? It actually took a while to figure this out, but here we know what LEP2 is. It's a gene which is called a macorin. It's a protein, it makes a protein. This protein is also present in humans and in flies and even in rice, so it's in all eukaryotes. And it has a particular organization of protein motifs. There are some zinc fingers, and they flank this so-called ring domain. So zinc fingers, maybe you remember that, they are usually used to bind nucleic acids like DNA or RNA, zinc finger transcription factors, right? And ring domains, ring stands strangely for really interesting new gene. And that is a protein interaction domain. People have studied these macorins a little bit in vertebrates, and I'm going to get to this later. We have these cool mutants. We have a whole bunch of mutants. So these are our different alleles of this. There are 2 isoforms, but then we have these mutants, and one removes the ring domain, but many of those zinc fingers are there, and that has the same phenotype as these point mutations, which are mutating only one nucleotide in the first zinc finger. So both of those domains must play a role in the function of this. And what about the next gene? What time is it? The other gene, LEP5, is more mysterious. In C. elegans, we have the entire genome available, and people have already looked at what might be a gene. There's also a transcriptome available, so we know which genes are being present or which gene products are present as RNAs. And this one got a provisionary name, H36L18.2. And it's on the X chromosome of these worms. And we have 2 alleles. And one of those alleles is a very large deletion, which removes the entire gene. No wonder that has a phenotype. The other one is a kind of weird change. It's just a point mutation. It's a G to A change, and it's at the very beginning of the gene. So now there's something special about nematodes. About 80% of their genes, when they are transcribed, there is a different sequence being attached to the 5' end. So that is called an SL1 sequence, splice leader 1. Most genes in C. elegans do that. So this is right at the beginning where the splice thing is attached. Otherwise, that gene is like any other. It has like this SL1 transplice, as I just said, that is pretty normal in nematodes. It's polyadenylated, so it could be anything. The transcript is fairly short, it's 934 nucleotides long, and people who were trying to find out where the genes are, even if we don't know what they really do, they annotated a protein. So they usually annotate a protein because, remember, we all think, or we used to think, that every gene is making a protein. So there's 83 amino acid long open read frame predicted. Wormbase is that's a database for worm people. There's everything which we ever want to know about genetics of C. elegans is going to be in Wormbase. The function is unknown, and that is because there's absolutely no indication that this H36L18.2 or LAB5 exists elsewhere outside of Caenorhabditis. So we didn't really know what this is, and we tried to find out whether that actually is a protein coding gene, and we find that it probably isn't, because I said it is present. It's not present outside of Caenorhabditis, but within Caenorhabditis it's extremely highly conserved, and this is what this image here is showing. It compares the sequence of 4 Caenorhabditis species, and the bigger these blue boxes, the more identical are the nucleotides. So you find a lot of blue here and a lot of blue here. Actually, more than in this region. This red thing here is where that supposed open reading frame is. So this is extremely highly conserved, but mostly on the side, on the ends of it, which would be 3' UTR and 5' UTR. But what is even more strange is that if we're looking at these other worms, so we have a highly conserved nucleotide sequence, but there's no conserved protein. So there's something annotated in C. elegans, but the same thing is not present in C. brixi, the other Caenorhabditis species, or Imani or whatever. We looked for small peptides. Maybe it's just a small peptide which this gene is gonna make, but there's none of this either. So it looks as if this actually doesn't encode a protein, but that was just all in silico here, so we wanted to show that. So we were using our mutant, and we were trying to, by inserting copies of this gene, to rescue the mutant, to make the mutant phenotype go away. This works if we are using the version of C. elegans. We just take a PCR product actually of C. elegans and put it into the worm, and it's going to rescue the mutant phenotype. So you have to explain these bars. So the red means that these worms, the males, we look at the adult males, right? They have a long lap tail tip. Most of them do. If I'm injecting into those worms a PCR product which comes from C. elegans wild type animals, this long lap tail goes away, and most of them look normal. That's the yellow. Now, if I'm taking the C. brixi sequence, and remember, there's no conserved protein at all. It rescues just as well, actually a little bit better even. So that blue here means that they show this over-retracted phenotype. It means that these worms actually start to retract their tail too early, because this is a heterochronic gene. If you put a lot of this, then apparently it makes tail tip retraction start earlier and earlier. And then, to be entirely sure, we introduced 2 stop codons, actually 2 and 3 stop codons into this annotated protein, and that didn't make any difference. It also rescues the phenotype. So we are concluding that this works as an RNA. So it's not a protein. It's going to work as this RNA. So now I already told you about microRNAs. There was the RNA revolution, so there we expect lots of microRNAs. So maybe that's another microRNA. The transcript is actually turned into little pieces. The problem is that we don't find this. So here is a picture of reads of RNA in different stages of C. elegans. So people have sequenced the entire transcriptome, and we can see that there is this RNA is present. This is our gene, so we find this present at very high levels. And if it was a precursor for a microRNA, we would expect that the precursor is not seen all that much. We would find the microRNA instead. So there are our colleagues, Victor Ambros, who is actually really famous for having discovered these first microRNAs. He has a data set where he did this with a special focus on microRNAs, and he cannot find this at all. So we think that this Lab5 actually acts as a long non-coding RNA, as the whole length of this thing. And the whole length of the thing. So we wanted to know whether we can figure out what it's actually doing. So we used computer programs to calculate the secondary structure. So that's an approximation. These things probably have a tertiary structure also. There's going to have, like, it will not be flat, right? But current computer programs can only do secondary structures, and it's already pretty informative. So we were using information from many species, and all of those dots here show, when they are black, that this is conserved across 15 actually Caenorhabditis species. So it seems as if these stem loops here could be important, and we wanted to make sure that that's the case. We have an indication that this stem loop is really important, because this is where that mutation was. I told you there's one weird point mutation at the beginning of that splice junction, and that's where this is. And I can now just put that mutation into the computer program and see what happens when we reconstruct the secondary structure. And it looks totally different. So if that now also happens under natural conditions, you modify this here to an A, then it would explain why this thing is not functioning anymore, because we may need this arm here. And so one way of figuring this out is you can recreate that structure by including a compensatory change, right? I can change this nucleotide and that one, and I did this here, so I changed the C30, which is the one which it binds to here, to a U, and that recreates the structure. So that's all happening in silico. We did all of this computer experiments. So does that mean anything in real life? We wanted to try that. Now we can do this same experiment also in our animals. We can make these constructs. We have already the mutant. This is same rescue experiments again. So if we don't do anything, then most of these worms have long lab tails, right? We already know that if we are putting the wild type version in, it rescues. The wild type version would have this structure. If we are putting the mutant version, there's a little bit of rescue. And we think this mutation actually doesn't cause it's not a null phenotype, so it's not completely gone, but it doesn't work as well. And I think this is what that shows pretty well. It doesn't work as well. And then I made a PCR product where I changed this other nucleotide also, so that we are getting the structure back. And as you can see here, it rescues again. So apparently the secondary structure of this really matters, and that arm here may be really important. Unfortunately, let me see, where are we here? This is where we are at this right now, so I do not really know what it is doing with this arm, but we can try to figure out what is this gene doing at all. So I only told you earlier that it somehow regulates negatively Lin28. Right? So here, both of those genes are actually negatively regulating Lin28, and it was kind of interesting that both of those genes although they are so different things. One is a protein, the other one is this long non-coding RNA. They have exactly the same phenotype. They're both at this place in the heterochronic pathway. So we wanted to know what it is actually doing to Lin28. And the first thing we did was look what happens to Lin28 protein. So this is, we can introduce into these worms a Lin28, which is tagged to GFP, so we can see the protein in these animals. And you can see what happens. It's really bright in first larval stages. I told you Lin28 is up in the beginning. It has to go away later so that LET7 can come up. And that's what's happening. So at L4s, when LET7 really needs to do its job, Lin28 is not there anymore, which is it's necessary. However, in these mutants, this here is LEP2. If we're looking at L4s, this Lin28 is still there. So apparently in these mutants, Lin28 protein is elevated. And one can do that. So this was a reporter, so we were looking whether the endogenous Lin28, the Lin28 which these animals make without us adding something to it, is also altered. This here is from a paper of some people who studied the Lin28 levels in quite a bit of detail with an antibody. So here is the antibody staining with Lin28. These are L1 animals. You can see that there's a lot of Lin28. It goes down until at the L3 stage there's very little left. This is a pretty dirty antibody, so there's some other stuff here. So even in the non-mutant there's a little bit here, but Lin28 goes away by the L3 stage. So we used that same antibody and we were looking at L3 animals where Lin28 shouldn't be there. So in our wild type animals there's not much left, maybe a little bit here, a little bit here, but in our mutants, this is a LEP2 mutant, here's a LEP5 mutant, there's still a lot of Lin28. So apparently both of those make that there's more Lin28. So now that could be because there's more messenger RNA, right? Lin28 has to be transcribed, so maybe they have just a lot more Lin28 messenger RNA, which would then explain why there's more protein. So we looked for this. We looked at what happens to the messenger RNA. The messenger RNA, there's a lot of it, actually not all that much, but there's quite a bit here, and then it goes down. So the way we are doing this, we are using quantitative PCR. So we are should I explain that? Maybe I won't. Anyway, so we are looking at the levels of this, and there's a 6-fold drop between the L1 and the L3 stage. And if we are looking at our mutants, we see that that's the same. So now they have a lot more protein, but they do not have any more messenger RNA. So somehow these two genes must affect Lin28 at a different level. I'm going to skip this slide. So actually I can't skip this slide. So I'm going to here we see this again, the Lin28 messenger RNA levels, they go down a little bit, but the protein levels, they drop a lot more. This is the blue line here. So we are thinking that somehow our two genes must have something to do with this protein going down even more. So this is from somebody else's work. They measured protein and messenger RNA levels very detailed. We think that these two genes have something to do with the downregulation of the protein, and we were wondering whether that is happening at the translational level, so it could be just not translated as well, or it could be that the already present Lin28, which, you know, there's Lin28 protein in the cells, that has to go away also, that they have something to do with protein degradation. So now there's a really cool tool which was recently developed, how one can distinguish between these two possibilities. That tool is called Dendra2. It's a fluorescent protein which is normally green, but when one shines short wavelength fluorescent light at this protein. It's going to change its conformation irreversibly, and it will then fluoresce red. So we did this. We made a construct. We fused this dendrite-2 to Lin-28. We expressed it in our worms. We saw that before. There's like bright expression of Lin-28. This is actually in neurons, where it's the brightest. And then we did this. We looked at this area here. Then we exposed it to red light. As you can see here, the green goes away. Not red light, to short wavelength light. And then it fluoresces red. So we did this in wild type and in our mutant. So now, what would happen? What do we expect is going to happen to this protein? So here we have wild type animals. This protein is probably going to go away. This is an L2 animal. When we look later, this would go away. There's no more translation because we know that Lin-28 is gone in later stages. In our mutant, however, we could have two possibilities. If translation is affected, then we will see new green because the old Lin-28 is now red. Or if degradation is affected, then the red would not disappear. So now let's see what happened. This is, we take this worm, put it back on a plate, let it eat for 24 hours and develop some more. So after these 24 hours, these animals are L4s. In our wild type animals, as expected, there is no more fluorescence or hardly any more fluorescence because Lin-28 is efficiently degraded. That should happen at this stage. In our mutant animal, they are still bright red. If they are still bright red, that means that somehow this red has not been degraded. The old Lin-28, which was present already here, is still around. And then there is still some green here, but as you can see, this photoconversion in this case was not 100%, so we think that this is also old green Lin-28. So this was done in lab 2, and we did the same experiment in lab 5, and we see exactly the same thing. So in both of those, the Lin-28 is not degraded properly. So this means that Lin-28, that both of those genes promote the degradation of Lin-28. So now, can we say anything about how these genes are actually doing this? How Macorin's, this is lab 2, Macorin is doing it. And here we are we are at the point where we have to do more experiment in the lab, but we can see what we know about Macorins from other organisms. So in humans and in mice, lab 2, no, not lab 2, Macorins have been shown to work as E3 ubiquitin ligases. That ring domain, the really interesting new gene domain, is actually the specific domain for which is found in these E3 ubiquitin ligases. So what are those? These are proteins which connect to another protein, the protein substrate, and they bring an E2 ubiquitin conjugating enzyme here. So and then this ubiquitin is being transferred to the substrate, and there can be a whole chain of these ubiquitin molecules attached to the substrate, and when that happens, then this is a message for the cell to degrade that protein. So it's being put into the proteasome as being degraded. So an E3 ubiquitin ligase makes this step here, the connection from this enzyme to the substrate specific. So you can imagine that Macorins are doing this. It binds to a Lin-28 and tags it for degradation. So we try to show this. We try to show that number one, Lin-28 is actually ubiquitinated, and that lab 2 somehow in our worms is tampering with this, and we couldn't so far. So maybe it's not working like that. And that is possible because remember there were these zinc fingers, and those zinc fingers usually don't have anything to do with the protein-protein interactions, but those were also really important. And as it turns out, in mammals, these macrorans have also been shown to have a function as RNA binding proteins. And so that is the other possibility, that macroran LAB2 is actually binding somehow to something else as an RNA binding protein. And whether these things function as E3 ubiquitin ligases or as RNA binding proteins seems to be context dependent. So now one context in which we know that this macroran plays a role is, interestingly, puberty. These people here found that mutations in human macrorans cause precocious puberty. So now suddenly macrorans, this weird gene LAB2, becomes another importance, I would say, because I think that, or we think that the macroran has to be put into this group of these genes which are conserved in all animals and probably play a role not just in worms but also in mammals and maybe in flies and other organisms. And the really good thing is that we can study this in worms at a genetic level which is totally impossible to do in mammals. So we're hoping that we're going to find out how these interact, and we think that that interaction may also play a role in mammals. So now what about LAB5? LAB5 was a long non-coding RNA, and I told you earlier about this RNA revolution. So after these small little RNAs were found, people were looking at the transcripts in organisms again, like for instance in humans, and they find that there's a lot of stuff is transcribed, and people were thinking that about 80% of what is transcribed is actually junk. So you know that if you look at the human genome, there are only so many genes. In between, there are long stretches which they were thinking is all just junk. But it turned out that a lot of this junk apparently plays a role. And as long RNAs, not just all as short RNAs, so it's not something new to have a long non-coding RNA. But what's known about these is that they act on transcription. They act in the nucleus. They have usually very low, they're lowly expressed, they're expressed in particular cells only, and they may somehow regulate transcription. So now let's look at LAB5. LAB5 actually is not lowly expressed. We find huge levels of LAB5. So this is a Lin28 messenger RNA, of which we know there's quite a bit, because Lin28 has to be made, and this is what we find for LAB5. And this is, so they're like more than 10 times as much LAB5, so it's not lowly expressed. And then we were trying to visualize this, so we were using a fluorescent probe, put it into the worms. The probe binds to the RNA of this LAB5, and we can visualize where it is, and you can see that this is a tail tip of an L2, so that's when there's a lot of this. You see there's a lot of it, but it's everywhere. It's not just in the nucleus. In fact, if you look here, you can kind of see this is where these nuclei are. It's excluded from the nucleus. So this is acting in the cytoplasm. And then if we were looking a little more broadly where it is, so these are neurons in the tail, and you can see that there is particularly bright expression of this here. And that is exactly what Lin28 protein is. So I think that this LAB5 is not working like these other long non-cutting RNAs for which we know already something. This must act in a different way, and we don't know yet. But to cite from this review about non-coding RNAs, they were asking, are all functions of non-coding RNAs known? And then they say, not a chance. So I think that we will be able to hopefully figure out what this is doing and add a new mechanism to what long non-coding RNAs are doing. So now, what did we learn? For us, it was really cool to find that the male tail tip is a great model to study the juvenile to adult transition. Because I showed you 3 retarded mutants, and none of these have any seam cell phenotype. So the tail tip adds really something to this. we found that macrons, which we discovered have a role in the heterochronic pathway in our worms, that they must be part of a conserved set of genes that regulate the timing of differentiation in animals. We know that it regulates the timing of the juvenile to adult transition in humans and in worms, but it's possible that it also acts in these other transitions. I said there's a transition between stem cells and differentiated cells, and also in cancer. So it's possible that macrons play a role there. People are still working on this in mammals. It's slow because it's either in mice or in stem cells. We are hoping that we will find out something out about this in our worms. So the third thing is that we discovered with Lab5 the first long non-coding RNA which has a role in development in C. elegans. So that's pretty cool. We don't know how it functions yet, but we think it functions in a different way from what we know about long non-coding RNAs. So we think that those functions are more diverse than we know already. And I think the other thing which we learned is that there's always more to learn. So we did a lot of work, and we have possibly more questions than we had before we started. And I think that's what research is about, and that's why it's really fun to do research, and it never gets boring. And I hope I didn't bore you guys, and I'm done here. Thank you for your attention.
Do we have any questions for Dr. Kiontke? Kelly? Are there any students first? All right, then we'll go we'll let you guys warm up while you're listening.
So presumably the LEP5 RNA is binding to something, a
Yes.
protein. Is there any evidence it might be binding directly to LIN28 and destabilizing the protein and can use an antibody to try to precipitate it down?
Yes,
Co-precipitate it?
we've been thinking about doing that experiment. This antibody was really horrible.
The LIN28.
So now there's a better antibody, and it may be better to do that experiment with this. So the other thing which we've been thinking is that it actually may bind to the macron, to LEP2. I have a pet hypothesis about how these things work, but it's kind of not fit for putting on a slide yet. But if LEP2 works as an RNA binding protein, then it could bind to a bunch of other messenger RNAs which are encoding proteins which will eventually degrade LIN28. And it would keep them in place until the right time. And then the right time would be when LEP5 is expressed, and then LEP5 somehow releases these all. So that is my pet hypothesis, but I have no evidence for this yet. Yeah, so but it could also bind directly to LIN28, but we still wouldn't know what it's doing then, because it would be a scaffolding kind of thing, so that it binds another protein. So we do know that there's a caspase which is acting on LIN28, but yeah, so.
We have a question over here, too.
Oh yeah.
So for LEP5, when you actually looked at all of the different worm species and you saw that there was a lot of conservation between them in terms of this particular sequence, presumably because it's so conserved, there's got to be some function in
Yes.
these other species. Did you actually take a look at each one of these other sequences from these other species and actually do sort of analyze that, run that through, and look at the secondary structure to see if they're all the same or very similar?
That secondary structure is convenient that it's still here. So how this works is this is the C. elegans structure, but it's being reconstructed based on the information from all of those 15 different species. And all of those dots which are black would show up if you do that analysis for any of the other species, because that is conserved. So you do this for all of them, and every black or the bright orange dots here are conserved. The structure is the same for all of those other species. And we tried with C. briggsii, which is like not the most distant species for which we could find this, but the C. briggsii sequence works perfectly well in C. elegans, so you can do that experiment. Yeah. Yeah, you can rescue the phenotype in C. elegans with the C. briggsii sequence. So now I don't know that sometimes people have done weird rescue experiments with more conserved genes where you put a fly gene into a worm or a mammalian gene. I don't know how well those experiments or what these experiments really tell you, but in this case I think it just means that this in all of those, this thing is going to be there. Also this area here actually, and this little bubble here. So these parts maybe not.
And so there's no homologue.
So
What
now
are they, so.
This is an RNA, and actually we were already pretty surprised that it's so highly conserved within this group of Caenorhabditis species. There is, so now Caenorhabditis, we now know of 50-something Caenorhabditis species. We have them all in culture. There is one clade which is more closely related to C. elegans. There they all have it. If you go outside of this clade, I couldn't find it. However, I think that is because it gets too divergent to see the homology. It's, you know, for protein coding genes, you always can look at the protein, and even if the nucleotides are really different, the protein may still be the same. But for this, it's an RNA, so we have to first find something to then do that again and see whether at least that little arm is there. So it may be only the structure. It may not have anything to do with what nucleotides are really leading to that structure. I mean, that experiment which we did is kind of indicative of this. Right? We modified 2 nucleotides here, and the nucleotides didn't matter, the structure did. Any more? Oh.
Oh, sorry.
Oh, there's this question back there.
Question
No?
back there?
No? You sure? But
You may have mentioned this, but so you're saying these genes are conserved in animals. Have you looked at to see, or are they present in unicellular or multicellular fungi?
So I have not looked at this. I can tell you that this macorin is present in plants. So I would think that it might be present in fungi, and I think now, I don't remember. LED7 is also very highly conserved. That is weird, actually. LED7 is an RNA, and that sequence is the same basically in us and in worms and everywhere. But I don't know about fungi. Sorry, got a blank there.
Right.
Is only the wild type able to reproduce, or all the mutants sterile or unable to reproduce?
Oh no, they are so. Fortunately, these guys are hermaphrodites, and the hermaphrodites are fine.
Right.
I mean, they have a shorter life than wild type animals because they do that extra molt, and that makes them very unstable. So they sometimes break open. The males of LEP2 are sterile. No, well, yeah. So they are not sterile. They don't mate. That's something which I didn't tell you. LEP2 males apparently don't know that they're adult. So they not like normal male would go to that partner and would eventually try to find where the vulva is and mate. These guys, they just walk around and then they bump into the hermaphrodite which you put on the plate for the mating test, and they don't do anything. And we think that the nervous system is not adult. So they behave like juveniles. They are not interested in sex. So, but and LEP5 males are fine. So yeah, and that tail tip, you know, it's kind of weird. They have that thing sticking out there, and in these guys and in the LEP5 males, it doesn't make any difference. So it doesn't hurt.
I have one quick question.
Sure.
I'll finish unless there's just a question from students. And my question is, so the LEP2 and LEP5, they're both loss of function mutants, and they result in retarded phenotype, right?
Yes.
That's what you call it. So the macorin mutant in humans was a precocious.
Yes.
So
You picked that up, right?
sorry. I mean, was
No,
that
yeah,
a
yeah.
loss of function too? Or is it
People
un-
think it is a loss
Okay.
of function mutation, and that is one of the things where this beautiful picture is not so beautiful anymore, because it seems as if somehow the direction of that interaction must be different. But who knows what's in between them? But yeah, you're
But
totally
it's
right.
affecting timing. It's just affecting.
Yeah,
timing.
it affecting
Yeah.
timing, but it affects it weirdly in the opposite way. Whereas Lin28 is the same thing. So Lin28 is precocious. Actually, our guys are retired. Yeah. So Lin28 is precocious in worms and in humans. So yeah, so that's one of the things where we don't really know. We have talked to people who work in mammalian systems, and they weren't too much bothered by that. I mean, I am
Doesn't bother me
actually
either.
very bothered by
I
this.
know.
Yeah.
All right. So one more time, thank you, Dr. Kiontke.
Well, thank you.